Huh? Like my system, but I'm joining live now. I'm making it live now. You're joining. Yeah, it's already live. It's already live. Huh? Like my system, but I'm joining live now. I'm making it live now. Yeah, it's already live. It's already live. Huh? Like my system, but I'm joining live now. I'm making it live now. Yeah, it's already. So let me know if you can see my screen. Let me know if you can see my screen. Okay, so we are live currently. We are live. Please, if you are available. Okay, thank you very much. Thank you very much. So last time we talked about amino acids and proteins. So before we go into proteins properly, let's quickly do a little review on amino acids. My name is Raymond Ibe. I'm a PhD holder in biochemistry. So here I want to demystify amino acid and how they form proteins. Now, like we talked about in our previous class, or our previous live session, we talked about amino acid, how we can identify the 20 amino acids that are found in proteins. We have thousands of amino acids, but only 20 of them that are found in proteins. And there are criteria for the amino acid to be found, for any amino acid to be found in any protein. So we'll also talk about the structure of the common amino acids. Talked about the essential and non-essential amino acid, like in our previous debate on our WhatsApp platform. We talked about if bread and beans, which one have more protein. So I was laughing at the comments, and I was just laughing, like, though we tried as much as possible to give different meaning, but I noticed some people were using AI to give their answers. So we also talk about essential and non-essential amino acids. So before we talk about a protein being a good, maybe a food being a good source of protein, we have to consider if the protein or if the food is derived, if the protein in the food is derived with essential amino acids, those ones that our body cannot produce. So we definitely need to get them from our diet. So we also talk about the arrow group, which are the classifiers. Arrow group classifies the individual amino acids that are found in proteins. Every amino acid that are found in proteins have different arrow groups. And it is the arrow group that confers some chemical and physical properties to the entire structure of a protein. So we also look at protein placement, how we can actually predict the location of an amino acid in a protein, either an integral protein, a peripheral protein, an anchor protein, for those of us that are in biochemistry. So I'll try as much as possible to also give more foundation to people that are not in biochemistry so that actually follow us through in this lecture. So we also talk about protein structure, how we can actually draw the structure of protein, even though we know that protein is a macro molecule. When we say macro molecule, that means the molecule is a large molecule. So this is an amino acid. You can see this amino acid have this amino acid has the side chain, which is the arrow group, which is a classifier that classifies each amino acid that is found in the protein. Now, every amino acid will have the carboxylic group. This is a carboxylic group, the COOH. It will also have this, what makes us call it an acid. It also have an amino group, which makes us call it amino. They will call it amino acid because of the amino group and the acidic group. So the amino group, the carboxylic group, and the hydrogen is common to every amino acid. It's common to every amino acid. The only difference between amino acid and amino acid is the side chain. So here, if we have hydrogen on the side chain, we refer that molecule as that amino acid as glycine. Refer to it as glycine. If we have CH2, COOH, like what you're seeing here, we refer to it as aspartic acid or aspartate. Aspartic acid or aspartate. It is called aspartate when it has lost this hydrogen atom. Call it aspartate. But with the presence of this hydrogen here, you call it aspartic acid. We have the one that has CH2 and the benzene ring. We call it phenylalanine. Call it phenylalanine. So now they say the alpha carbon atom in amino acid are chiral centers except for glycine. The implication of that is that glycine is optically inactive, and there's what we call optical activity. A molecule that, if we say a molecule is optically active, the molecule should be able to rotate a light, a plane of polarized light, either to the left or to the right. So that's why we see D-glucose, L-glucose, D-alanine, L-alanine. The D there tells you that, oh, this molecule can actually rotate a plane of polarized light to the right. If it is L, you can say, okay, this molecule can rotate a plane of polarized light to the left. Example. Okay, you can see this is light. This is the polarizer. Then we have the sample tube, the container that contains your sample, either D-glucose or L-glucose. You can see the angle of the analyzer. So if the light source is coming, this is a white light, so it has actually different light rays. So the light will come into the polarizer. The polarizer will streamline the light into one particular light, which is the polarized light. The polarizer hits the tube that contains the molecule. You can see the polarizer going to the right. You can see it's actually having an angle of plus 45 degrees. That tells you more about that molecule. So that tells you can see the polarization going towards the right. That tells you that whatever molecule that is present in that chamber is rotating the light to the right. So that molecule will be called a D molecule. So you can actually add D to the name, which will not say, if it is glucose, we'll say it is D-glucose. If it is mannose, we'll say it is D-mannose. If it's an amino acid like aspartate, we'll say it is D-aspartate or D-alanine. So here we have, we talked about the arrow group as the classifier. So we use arrow group to classify the molecule, the amino acid that are found in proteins. The amino acid that are found in proteins. So for here, we have the nonpolar hydrophobic arrow group. So here we talk about the arrow group that are hydrophobic. That means they don't like water. They're hydrophobic, they don't like water. So if you put the amino acid in water, they will try as much as possible to withdraw, or we call it to sequester away from the aqueous environment. So we call them hydrophobic, non-polar. You can see here we have hydrogen for glycine. We have hydrogen for glycine. We have CH3 for alanine. We have, you can see this group for proline. We can see valine. We can see isoleucine. We can see leucine. We can see isoleucine and methionine. You can see isoleucine and methionine. So these amino acids are called hydrophobic or nonpolar amino acids. Say, if these amino acids are found in a protein, you know, we say protein is a polymer made up of monomers of amino acid. So these molecules, this amino acid they are seeing here, they are the ones that are joined together to make what is called a protein. So if these molecules if these amino acids are found in a protein, we always tell you that they will be found within the protein that is, if it's an integral protein, it will be found within the portion of the protein that is shying away from water. You will see them, they will cause the protein to fold in such a way that they will be shying away from water environment. They will be moving away from the water environment. And there are simple ways you can actually draw these amino acids. You can actually draw them. You can actually draw them. So just look at their common features. If you look at the common features, you can actually draw them. For glycines, just is only one hydrogen. You're done with glycine. For alanine, just add CH3. You're done with alanine. The alanine and phenylalanine are almost the same. In phenylalanine, you remove one of the hydro, which is my remaining two hydrogen, and draw a benzene ring. You will get a phenylalanine. But I will get into that. We'll go into that, how we can actually draw the structures easily. So we have the aromatic arrow groups. These are the arrow groups that contains aromatic rings, or phenyl rings, or benzyl or benzene rings. So we have phenylalanine, thyroxine, and tryptophan. These are the three amino acids that are aromatic in nature. And they're aromatic because they contain the benzene ring. This is the benzene ring. You can see this molecule also has a benzene ring. This one also has a what? A benzene ring. So we call them aromatic amino acid. Now, one of the characteristics of this amino acid is that they absorb light strongly at 280 nanometer. And they are relatively non-polar. When we say relatively non-polar, it means they can also have some polar group that can make them have slightly polarity and more of hydrophobicity. Now you can see for thyroxine, thyroxine has OH. Thyroxine has OH. So that OH there is conferring hydrophilicity properties to the thyroxine. Then you can see for there's an amino group here for tryptophan, which can also confer some hydrophilicity property to tryptophan. But generally, they are hydrophobic in nature. Now, thyroxine kinases phosphorylate the hydroxy group. When we go further in biochemistry or in whatever endeavor, we'll start understanding why they are called thyroxine kinases. They are called thyroxine kinases because these are enzymes. When we talk about kinase, talk about enzymes. These are enzymes that can phosphorylate a protein at a thyroxine residue. Remember, I told you that these are monomers that makes up a polymer called proteins. So now this thyroxine can be found in the protein. So if thyroxine is found in the protein, the enzyme that can phosphorylate that protein, because some enzymes are active upon phosphorylation. So this tyrosine kinase is to phosphorylate those enzymes or those proteins, activating those proteins. And they can only phosphorylate the protein at the thyroxine residue. That's why they are called thyroxine kinases. So the kinase there, what means that they are phosphorylating they are using energy for their phosphorylation. They are using what? Energy from their phosphorylation. And the energy source is ATP. Adenosine triphosphate. The energy source is ATP. So thyroxine and tryptophan are slightly hydrophilic, which I've actually explained what confers the hydrophilicity properties to these amino acids. We also have positively charged slash basic amino acids. Somebody is saying it's looking blurry. I can barely see. Okay. Let me see if I can actually increase the. Okay. Is it better? Okay, I think I can actually zoom in. Is it better? I can actually zoom in so that I will see better. Okay, so I can actually zoom in. Okay, so we have the amino acids that are negatively charged. Okay, so we have the amino acids that are negatively charged. So these are negatively charged arrow groups. So these negatively charged arrow groups have the negative group of, we have, there are only two of them. We have, what's it called? We have glutamates. You can see the CO minus in glutamate. You have your glutamate, you have your aspartate. These are the negatively charged amino acid. So you can see that they have CO minus at the terminal. So now, if you know the structure of glutamate, you can actually draw the structure of aspartate. Just for aspartate, you have CH2CO minus. For glutamate, just add additional CH2 and you're done with glutamate. So whatever, if you can draw the structure of aspartate, you can actually draw the structure of glutamate. Just increase the carbon length by CH2, you have glutamate. And these are the only two acidic amino acids. They are acidic in nature because of the CO minus. So that's the acidity there. So if the H is present instead of the minus, if you can put the H here, if you can put the H here, it will become aspartic acid. If you put the H here, it becomes glutamic acid. But removal of the H will make it aspartate or glutamate. We also have the positively charged arrow groups. The positively charged arrow groups. You can see that the only difference is the arrow groups. So for lysine, you notice that we have how many CH2? 1, 2, 3, 4. 4 CH2 with one amino group. You're done with lysine. For arginine, you can see we have how many CH? 1, 2, 3. Then we have the amino group here. So that's for arginine. We also have histidine. We also have histidine. Also have histidine. So these 3 amino acids here are called positively charged or basic arrow group. They are called basic because they behave like a base. They are called basic because they behave like a base and they are positively charged. So, but we also know that they are also hydrophilic in nature. They are also hydrophilic in nature, so they can interact well with water. The negatively charged arrow groups or acidic groups are also they are also polar, which means they are also hydrophilic in nature. They're also hydrophilic in nature. So, and if they are found in the protein, it's not their character will not tell you the location where they are likely to be found in that protein and how they can contribute to the overall structure of the protein. So here we have polar hydrophilic uncharged arrow groups. Uncharged, remember the other ones were charged. That's why they have that's why they are positively charged or negatively charged. So here we have the polar uncharged arrow group. It means this one they are polar, which means they can dissolve well in water, but they are not charged. They are neutral in nature. And there are properties that confers that hydrophilicity to them. You can see the OH in circle. Hydroxyl group makes it hydrophilic. You can see for threonine, you can see if you draw the structure of serine, which is CH2OH only, you just add another CH3 and you get threonine. If you remove the OH and add SH, you have cysteine. For the other one we have here is asparagine and glutamine. You can, if you look at the structure of asparagine and glutamine, you find out that it has a similar structure to asparagine has a similar structure to aspartate. Glutamine has a similar structure to what? Glutamine has a similar structure to glutamate. In fact, sometimes they are referred to as the amine nature, the amine counterparts of aspartic and glutamic acid. You can see asparagine and glutamate. For glutamate, there's a replacement of this NH2 with oxygen as aspartate. For glutamate here, there is a replacement of this NH2 with oxygen. That makes it glutamate and aspartate. If we replace the oxygen with NH2, we have asparagine and glutamine. So these are the 20 amino acids that are found in proteins. So when we go into protein properly, we now start saying, you know, their functional group, which is the arrow group, into play. So we have this. These are uncommon amino acids. Uncommon amino acids. We have the hydroxyproline, 5-hydroxyl lysine. These are 2 important amino acids that are found in collagen. Usually for those of you that are into skincare, you know, skin products, they always tell you that this cream is rich in collagen. This cream is, they would tell you something like collagen in order to sell the product. But the truth is that we know that collagen is a protein. So what we believe is that in biochemistry, if you're deficient in vitamin C, you have the disease called scurvy, which is characterized by fragile arteries, bleeding gums. When you brush your teeth in the morning, you notice that you're bleeding, means that your vitamin C is not, you're lacking vitamin C, or your collagen are not strong, which might be as a result of deficiency of vitamin C or the amino acids that are important in synthesizing collagen. And one of the amino acids that is important in synthesizing collagen is hydroxy, is proline and lysine. Now, these are two amino acids that are done by post-translational modification. Usually, what we mean by translational modification is that the protein will be synthesized. After synthesis, the enzyme will not start forming a hydroxy group of the proline and lysine residue. And those enzymes are, vitamin C is important for those enzymes activity. So if vitamin C is not present, the enzyme will not be fully active to convert proline and lysine to 4-hydroxyproline and 5-hydroxy lysine. So that's what they'll tell you. Make sure your vitamin C, you're not deficient of vitamin C. And that's why most of those creams are also rich in vitamin C, because they know that vitamin C is responsible or is implicated in the synthesis of collagen. So we have, these are uncommon amino acids. We have the selenocysteine, we have the desmosine, we have the 6N methyllysine. So these are the essential and non-essential amino acids. For the essential amino acid, we have histidine, isoleucine, leucine, lysine, methionine, phenylalanine, threonine, tryptophan, and valine. For the non-essential, we have alanine, arginine, asparagine, aspartic acid, cysteine, glutamic acid, glutamine, glycine, proline, serine, and thyroxine. You can see that lysine, which is implicated in the synthesis of collagen for your strong skin, for your skin tone, is actually an essential amino acid. So if your diet or if your food is not rich in lysine as an amino acid, as an essential amino acid, you might not be synthesizing your collagen very well. You can even start having those stretch marks and all those things. They usually, those skin breaking that we see in dry and flaky skins. So we have, you can see proline is not essential. So even though your body can produce proline, your body still needs lysine for the synthesis of collagen. And if you're deficient in lysine, you'll not be producing your collagen properly. And it will result in some of the symptoms that are associated with inadequate production of collagen. We also have these amino acids that are called conditionally essential amino acid. These are amino acids that are, maybe their precursors are inadequate. What I mean by precursors, that what the body uses to produce them are not there. They become conditionally essential. Or in a disease state, maybe you're having a disease that your body cannot produce the enzymes that are necessary for the production of those amino acids. Those amino acids becomes a conditionally essential amino acid. So you have to take them via diet. So histidine was once thought, let me increase the, okay. So you can see histidine was once thought to be essential only for infants. But now it is known that even small amounts are also needed for adults. So even though your body can produce insulin, you also need histidine in your diet, small amount of insulin in your diet. So now we are in proteins proper. So what are proteins? I need people to start making comments so that I can know if you're actually following. I need people to start making comments, like comments. Let me see. Are you with me? Are we following? I want to know if we are following. Okay, yes. We're actually following. So proteins are large, complex biomolecules made up of chains or smaller units called amino acids. So you can see the importance of the amino acid, which are linked together by peptide bond. They are essential macromolecules in all living organisms and play diverse roles or diverse functional roles in the biological system. So there are thousands of roles of proteins in the biological system. You can see currently because you have proteins in your eyes. You can digest your food because of proteins. You can actually send information in your body because of proteins. You can actually fight foreign organisms, foreign microorganisms because of proteins. You can actually fight disease because of proteins. You can actually breathe very well because of proteins. So proteins are essentially important. In fact, there are some proteins that are essential to life. If those proteins are not properly synthesized, the individual will be at risk. For example, the protein that is responsible for transportation of oxygen, which is hemoglobin. If your hemoglobin is bad or deficient, you definitely have problem. As we see in sickle cell disease, they have hemoglobin, the same way we have hemoglobin, but because of a change in amino acid in hemoglobin structure, there is a change in the function of the protein. Okay, somebody asked, I would love to know the food that are rich in lysine. Of course, any animal protein, will be rich in lysine. Even egg is rich in lysine. They usually call them first class proteins. So these are different functions of protein. Protein can actually act as a structural component of the system. Example, like you see in collagen, like we've talked earlier, even keratin, which is the hair. Your hair is actually protein too. We also talk about enzymes. Proteins are, most of the enzymes in biological systems are proteins, catalyzing biochemical reactions. We also talk about transport, which is hemoglobin. We also talk about defense system. We also talk about hormones like insulin and glucagon. They are protein hormones that regulate glucose homeostasis, how your body regulates glucose. And that is why if your body can no longer produce insulin, or your body can no longer sense insulin or receptive to insulin, we talk about diabetes. So you can imagine that if there's a problem in your insulin, diabetes sets in. If there is a problem in your glucagon, diabetes sets in. So insulin is actually a principal hormone in blood glucose regulation. Now, glucagon is actually also an hormone that is responsible for making sure that the stored glucose in your liver or in your kidney is being pushed out into the blood whenever you're fasting. Whenever you're fasting, they store the glucose in your liver and your kidney. It's pushed out to the blood so that glucose can be supplied to other cells or organ that rely majorly on glucose. So when we go into metabolism, we'll do that properly. So this is the building blocks of protein. We have amino acids coming together, two amino acids coming together as what you can see here. Two amino acids coming together as what you can see here. So we have this amino acid. You can see that this arrow group can be lysine, arginine, leucine, isoleucine, glycine, methionine, or anything. This arrow group here can also be any other amino acid. But the most important thing is that the two amino acids are coming together. One is donating the carboxylic end, one is donating the amino end. So the carboxylic end and the amino end coming together forms this, what is called the peptide bond, with the removal of water, which is H2O. Removal of water. That is why before we cook our meat, we have to add water. You have to add water because water makes tenderize the meat, which by hydrolysis, it will break down most of the peptide bond, making the meat accessible for consumption. Okay, somebody asks, see, I also mentioned conditional essential amino acid is histidine, the only exemption of such. Okay. Yes, I mentioned conditional essential amino acid, and those amino acids that maybe what the building blocks or the molecules that are needed for their production is not available. You have to not take them through diet. You have to not take them through diet. So when they form together, they form the peptide bond. So now this is just two amino acids. So it will be called a dipeptide. If we have another amino acid joined to this hydroxy group here, it becomes tripeptide. If we have another one joined together, it becomes tetrapeptide. If we have 5, it becomes pentapeptide. If we have so many, it becomes a polypeptide, or which is called, either I'll call it the polypeptide or call it proteins. Call it polypeptide or call it the proteins. So now we have levels of protein organization. Levels of protein organization. These are 4 levels of protein organization. We have the primary structure, which is just the sequence of the amino acids that are found in the protein. So if you look at the protein, all the amino acid, their sequence of arrangement is defined as the primary structure of that protein. So we have the secondary structure. In secondary structure, we talk about interactions between amino acid residues in the protein. Usually we talk about interaction between 4 amino acid residues apart. So either by hydrogen bonding or by any kind of interaction. So if there's an interaction between 4 amino acids apart, it will create a kind of structure which you term the secondary structure. And this secondary structure can actually be a primary, the alpha helical structure or the beta plated sheets. The alpha helical structure would look like this protein on the screen. This is alpha helix. So we also have the proteins that can look like the beta sheets. And there are amino acids that when they are found within the particular region of the protein, it will turn the protein to start having the secondary type of beta sheet. Example is glycine and proline. These are 2 amino acids that can change the conformation of the protein and makes it look like a beta plated sheet. Okay, I said insulin is the principal hormone in blood glucose homeostasis. What is homeostasis? Homeostasis is the regulation of a fairly internal constant fairly internal constant environment. So when we look at glucose, glucose is maintained within a threshold in the blood. So an insulin is responsible for maintaining, insulin and glucagon is responsible for maintaining that threshold, such that if the body cannot maintain that threshold and there's increase in blood glucose, we term it hyperglycemia. If it is low, we term it hypoglycemia. Now, if it is consistency higher, which is consistency in hyperglycemia, we say the individual has a diabetes. So the tertiary structure is when we have two secondary structures, two or more secondary structures present in a protein. We can have two alpha helix joined together, like you can see in this board now on the screen. You can see the alpha helix in green ribbon. You have 1, 2, 3. You have ones in blue ribbon, right? So the blue, you can see there are almost about 6 of them, 6 different alpha helix joined together. So that is actually called a tertiary structure. 2 or more secondary structures linked together. So that's what we'll talk about the tertiary structure. But quaternary structure is when you have 2 or more tertiary structure covalently linked. 2 or more secondary structure and 2 or more tertiary structure that are covalently, and I said that, not sorry, 2 or more structure that are non-covalently, please, non-covalently linked. If they are covalently linked, it is the same structure. We'll refer to them as a tertiary structure. But if the linkage between them is non-covalent, we'll say they are quaternary structure. I'll give an example with insulin. With insulin. And that will be, okay, let me see if I can do that at this point. Let me see if I can do that. Insulin is made up of 2 chains, chain A and chain B. Chain A and chain B. Now, at chain B, we have sulfur amino acid, which is cysteine. Anytime you see anything like that, know that it is cysteine that is present there, because cysteine is the only sulfur containing amino acid that has a free hydrogen. Methionine doesn't have, so it cannot be methionine. So we have sulfur. So we have another sulfur linking 2 of them here. So the chain A has sulfur unit cysteine. The chain B has also cysteine. So when the 2 of them are linked together, this becomes one protein, because the sulfur linkage is a covalent linkage. So even though there are 2 chains, but because of they are covalently linked, it is termed one protein, and this is a tertiary protein. So we can actually separate them into 2 different chain A and chain B, but that doesn't mean that they are not, if they are joined together, they can be joined together by a covalent linkage, and that makes them one protein. So I've explained the different structural design of protein, but let's actually look at the titration curve of this amino acid. We say this amino acid, they have both acidic and basic properties. So now they have 2 pH region, 2 pH space, or 2 pH points. Now we have the PK1 and the PK2. Now the PK1 is where we talk about proteination, and it is very important that you understand proteination and deproteination. Now, the importance of this proteination and deproteination is what we understood when we talk about the naturation of protein in pH solution. For example, we know that the stomach has an acidic pH of 1 to 2. So every protein or every enzyme that's present. Okay, let me increase this. I need your responses so that I can be able to. I need your responses so that I can be able to adjust whenever it is necessary. So now this pK1, which is 2.34, is actually the pH at which the carboxylic group will donate its own hydrogen. Now, the pi you're seeing here, the pi you're seeing there is called the isoelectric point. It's called the what? The isoelectric point. The isoelectric point is the pH at which there is no net charge in the amino acid. That means the amino acid has a net charge of 0. If you come up, you see that we have plus NH3 and COO minus. So you can see plus cancel minus. So they say the amino acid is neutral at that particular pH. Say the amino acid is neutral at that particular pH. So, but above that pH, as we are going above the pH, that pH is 5.97. So you can say as 5.97, the amino acid glycine is electrically neutral. But at pH of 9.6, the amino acid is negatively charged. The amino acid is negatively charged. So you can see that at acidic environments, of you can see the pH of between 0 to, let's say 0 to 4, 0 to 5. You can see that the glycine will be in this form. The glycine will be in this form. So that means at acidic pH, the amino acid glycine is positively charged. But as you go further, as you increase the pH, the amino acid becomes neutral. As you can see, it becomes what? Neutral. But as you go up to the basic region, you can see we are into about 8 to 9. You can see that the amino acid have donated its hydrogen. Even the amino group have donated its hydrogen. The amino group becomes neutral, but the carboxylic group becomes negatively charged. So at that pH, we say the amino acid is negatively charged. So there is an implication of this when they are present in the protein. There's an implication of this when they are present in the protein. But when they are present in the protein, it is not the COO minus and the NH, these are the parts that is forming that can interact with the environment. It is the arrow group. Mind you, the arrow group that contains COO minus will react. The arrow group that contains NH3 plus will also react. But the amino group and the carboxylic group that forms the bonding will not react. They can only react like this if they are present, if they are alone in a solution. But when they are in the presence of the protein, or when they form a protein, it is only the arrow group that react. And that is why we define them by the arrow group. So it is the arrow group that will now determine their chemical properties if they are found in the solution. So like what we are seeing here, in this case, we are trying to compare the pKa or the pK of acetic acid, which is an organic acid, and an amino acid. But yeah, there's a reason for, yeah, I said the pKa for the carboxylic group is 4.8. But the pKa of amino group, even though it's also a carboxylic group in amino acid, it has a reduced which is 2.34. So you can see that when they are present in amino acid, they easily give out their hydrogen. But when they are present, when they are not in an amino acid form, like you see in aspartates, in acetic acid, rather, they will hardly give out their hydrogen. And there's a reason for that. There's a reason for that. Because of the hydrogen here, there's a repulsion between the amino group and the departing proton. The departing proton is this proton here, which is the hydrogen, the COO, the COO. The hydrogen is the departing proton. So because of the repulsion, move away, please. Because of the hydrogen is positively charged, the hydrogen in the amino group is also positively charged. So they repel. So that repulsion will cause an easy breakage of the hydrogen. So that's why the pKa of the carboxylic group in amino acid is lower than when they are present in an ordinary acid. Just like when they say when two people are always quarreling, it will be easy to separate them because they are already in the repulsive energy. When people are very attracted to each other, they are not quarreling, they are peaceful at each other, it will be difficult for you to separate them. So that is what is happening in these amino acids. That's what is happening in this amino acid. So so the same thing happens with I want to see comments. I want to see comments, please. Let me see your comments. We are following. Let me see your comments. Let me see your comments if you are following. Are you with me? So you can see here we have glutamate. We have glutamate. So you can see in acidic solution, the first pKa is even lower because of the repulsive energy between the hydrogen of carboxylic group and the hydrogen of the amino group. Okay, I'm at the graph. Ask the question, please. I can use this graph to explain the previous graph. I can use this graph to explain the previous graph. So what we have here is glutamate. The first graph. Ask your question. Let me attend to it. Ask a question. Ask a question. Okay, this is the first graph. This is the first graph. So I want to get your question. You're not asking your question, okay? And whenever you ask a question, I'll proceed from there. Okay, yes, exactly the same thing happens with glutamate. You can see here the pKa is even reduced further. We have the pKr, which is the R means for the arrow group. So we have the pK2. You have the pK2. So you can see the different pH denotes the nature of the amino acid. You might be asked, at pH of this, what is the predominant form of the amino acid aspartate or the amino acid glutamate? So you should be able to know the predominant form of the amino acid in different pH concentration or in different pH. So we know that at acidic pH, the amino group is still retaining the positively charged group at lower pH. As the pH increases, the carboxylic group begin to lose their hydrogen. As the pH increases further to the basic region, the amino group loses its own hydrogen and make the carboxylic group is also still negatively charged. So you can see here, at pH of 9.67, the total charge of glutamate is -2. At pH of within that same pH, you can actually have below that pH, you can actually have or let's say at pH of 9.6, the predominant form is -1 because we have plus here and 2 minus. So 1 plus and 2 minus will give us +1 or -1 rather, -1. Okay, if you cannot see the lecture video will be posted here on YouTube, so you can actually go back and actually watch the lecture video. So while we progress. So for the next one, the same thing happens with histidine, just the same nature. Then we can look at this protein here. This protein is made of this peptide. This peptide is made up of 5 amino acid. 5 amino acid. The first one is serine. Second one is glycine. Third one is threonine. The fourth one is the third one is thyroxine. The fourth one is alanine. And the fifth one is what? Leucine. And the fifth one is leucine. So you can see that they use you can see the CH2OH, we talk about it as being serine. Only hydrogen, we talked about it as being glycine. CH2COO, CH with the benzoic and the hydroxy group says thyroxine. Thyroxine sometimes is also called para-hydroxyphenylalanine. When we'll talk about inborn errors of amino acid metabolism, I will explain the importance of this in an inborn error called phenylketonuria. We have CH3, which is alanine. We have CH2, CH, then CH3 and CH3. We have leucine. So this molecule you're seeing here is called aspartame. Aspartame is an artificial sweetener. It's a peptide. It's an artificial sweetener. Usually they use it in pediatric formulation to produce drugs for children, because usually children don't like things that are bitter. So they usually add this aspartame to sweeten the drug so that children can actually easily consume those drugs. But there's a side effect to this. There's a side effect to this. In fact, in the container, they will write in the container of the drug that this drug contains aspartame, a source of phenylalanine. A source of phenylalanine. So for children with phenylketonuria, cannot take that drug. That's why they are sounding that warning. Cannot take that drug because the aspartame contains phenylalanine, and phenylalanine is the problem for children with phenylketonuria, which is an inborn error of amino acid metabolism. So you can see here the aspartame contains aspartic acid. You can see aspartate contains phenylalanine and methanol. So it's called aspartame. So L-aspartyl phenylalanine methyl ester is an artificial sweetener that is found in even in your soft drinks. Where they will say this drink is zero sugar. It is aspartame that is the sweetener there. It's zero sugar. So those that drink zero soda, zero this, zero that, zero this, it is aspartame that you're drinking. Aspartame that you're drinking. So we have insulin, we have glucagon. These are biologically active peptides and polypeptides. We have the corticotropin. We have the oxytocin, which is implicated in uterine contraction for pregnant women when they go to labor room. This is what actually is responsible for contraction of the uterus for delivery. So we have subunit, multi-subunit proteins. These are proteins that have multi-subunits. So we have single proteins versus multi-subunit proteins. Some protein contains or consists, let me, some proteins contain some so you can see some proteins are single polypeptide chain. Most subunits have two or more subunits that are associated non-covalently. That was what I was saying earlier. The association must be non-covalent association if they should be called a tertiary protein, if they should be called a quaternary protein. So if two oligomeric protein, if two or more subunits are identical, they are called protomers. Example is hemoglobin. Hemoglobin has four subunits, two identical, two are non-identical, two identical, which is different from the other two. We have the alpha and the beta subunits. The alpha subunits are identical, two alpha subunits The beta units are also identical. So it's a dimer of alpha beta protomers. It's a dimer of alpha beta protomer, or we say is a tetramer, which is have 4 subunits. So if you say they are tetramers, you can say, okay, it has 4 subunits. But if you want to go further to indicate the nature of the subunits, if there is any subunit that are identical to any of the subunits, you cannot use the word protomer. A dimer of alpha beta. So alpha beta protomers. Protomers is alpha is 1, beta is 1. So dimer means 2. 2 alpha beta and 2 alpha subunits and 2 beta subunits. I don't know if you can see this. This actually, most times there are reasons to everything. Let me use that word. There are reasons to everything. Sometimes they will tell us, or most times, they will tell us the average molecular weight of an amino acid in a protein is 110. 110. 110. 110. So tell us that the average molecular weight of amino acid is 110 molecular weight or 110,000. 110 molecular weight or 110,000. Now, alanine has a molecular weight of 89. Let me enlarge my screen. Alanine has a molecular weight of 89. You can see what we say, free molecular weight and residue molecular weight. They are two different things. Free molecular weight and residue molecular weight. Free molecular weight is when they are not part of any protein. When they are not part of any protein, we say they are free. When they are part of the protein, we call it residue molecular weight. So you can see the molecular weight when it is free is 89. Why is it 71 when it is present in the protein? Who can tell us why? Who can tell us why it is 71.08 when it is present in the protein? Who can tell us why? I want to see comments. 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 Okay, somebody said because it loses either its carboxylic or amino group to form the peptide. Okay, I didn't join. Okay, so now if you subtract 18 from 89.9, what do we have? Subtract 18 from 89.09. We subtract 18, what do we have? We have 71. So where is the 18 coming from? 18 is coming from the water that have been lost, that water molecule. Remember, it's 2 hydrogen, which is 2 plus 16 of oxygen. That's why we have 18. So if you subtract 18 from each of them, you have the residue molecular weight. We have the residue molecular weight. Yes. So that is why when it is present in a residue, like when it's present in the protein, the molecular weight will reduce by 18. Now, if we add all of them, these are the 20 amino acids. These are the 20 amino acids. If we add all of them, we'll have 100 and we have if we add all the residue molecular weight, we'll have 2,377.74. If we divide it by 20, which is the number of amino acids that are found in the protein, we'll have 118. We have 118.88. So now the average molecular weight is 110. How did we get to 110? Why did we accept 110? Why did we accept 110? They accepted the 110 because studies, different protein studies, different protein structure determination of protein analysis have shown that the majority of the amino acids that are found in protein, listen to this carefully, the majority of the amino acids that are found in proteins are the small molecular weight amino acids. And the small molecular weight amino acid. That means if you have a protein that have up to 1,000 amino acid, 1,000 amino acid, 80% of the amino acid that will be found in that protein will be smaller molecular weight amino acid. Example, alanine, threonine, serine, cysteine, alanine, leucine, valine. So we do smaller molecular weight, and if you check those smaller molecular weight amino acid, you can see this one 99. 99 will be valine. 99 will be valine. You can see threonine is 101, serine is 89, proline is 97. Proline is 97. Alanine is 71. Alanine is 71. Aspartic acid 115. Cysteine is 103. Glycine is 57. Isoleucine and leucine are 113 and 113. So you see that, and these are the major amino acids that will be found in the protein. So since the majority of amino acids that are found in the protein are not even up to 110, if we add those smaller amino acids together, if we add those smaller amino acids, it will be approximating to 110. So we decided to accept 110 as the average molecular weight of residue amino acid. So that is what we get. That's what we get. That's what we get for something like this. I will explain this question later. I'll explain this question later. Okay, so now we also know that some proteins, contains other groups that are attached or chemically associated to the proteins. Some of these groups they are called conjugated proteins. They are proteins that have other groups attached to them. They are called conjugated proteins. The non-amino acid part of the conjugated protein is usually called the prosthetic group. The prosthetic groups are groups that are covalently linked or covalently associated with the protein. Example, we call lipoproteins. Lipoproteins are example, low density lipoprotein, cholesterol, high density lipoprotein, cholesterol. So we have the lipoprotein contains lipids and proteins. We have glycoprotein that contains sugars and proteins. We have the metalloproteins. These are proteins that contains the metal group. Example, potassium. Example, sodium. Example, iron, which is ferrous, that are found in hemoglobin. You understand me? So these are different conjugated proteins. But let's go into, okay, these are just the levels of protein structure: primary, the secondary, tertiary, and the quaternary structure of proteins. So now let me explain this. Let me explain this. Now, this is amino acid. This is bovine cytochrome C and bovine chemotrypsinogen. Now, alanine for bovine cytochrome is 6 in number. We have 6 alanine present in the protein. The entire number of the protein is 104 for cytochromes, bovine cytochrome C. For bovine chemotrypsinogen, we have total amino acid as 245 amino acid. So you can see in the 245 amino acid, we have more of threonine and serine and valine, even leucine, glycine. You can see that the smaller molecular weight of amino acid are forming the bulk of the protein. So that's why we agreed that the average molecular weight of amino acid in the protein should be 110. So if I give you that a protein with 20,000 Dalton or 20 kilodalton, what is the number of amino acid in the protein? You now divide the 20 Dalton, the 20 kilodalton, or the 20,000 Dalton divided by 110. It will give you the number of amino acids that are found in that protein. They have tests for protein. So now somebody might sell a product for you and tell you that this product is rich in proteins, while there is no even any protein in that product. Somebody might just say, this product, now bunge product, you know, how we do see it in Nigeria, how do we see it in this part of the world? They will tell you, enter bus, traveling to different places, you see people marketing different types of products. They might even tell you, this protein is rich, this supplement is rich in proteins. So you can actually take those supplements and test them to see if there's protein present. There's protein present in that supplement. So how do we test for protein? The major test for protein is the Biuret test. The Biuret test. The Biuret test detects peptide bond. Take note of that. Detect peptide bond. The Biuret detects peptide bond. So, and this, it actually occurs in alkaline solution. It actually occurs in alkaline solution. So in alkaline solution, the copper ion from copper salt or copper sulfate forms a coordination complex with the nitrogen atoms of the peptide bond. Each peptide bond provides a lone pairs from the nitrogen or the amino group that coordinates with copper. This produces a violet or purple colored complex. So you can see on the right, I have different test tubes. The test tube 1 is phenol. Test tube 2 is thyroxine. Test tube 3 is egg albumin. Test tube 4 is starch. And test tube 5 is distilled water. Now, if you put Biuret reagent is blue in color. If you put the Biuret test in all of them will shine blue. Don't mind the color you're seeing there. All of them will shine blue. The thyroxine will shine blue. The starch will shine blue. The distilled water will shine blue. But the only indication that there's protein present if the blue color changes to purple or violet color, that will not tell you that there is presence of protein. So thyroxine is an amino acid. It's an amino acid. It will not turn purple. It will not turn purple. It can only turn purple if there is a presence of a peptide bond. So thyroxine is an amino acid. It's not forming any peptide bond with another amino acid. So it will not react with the Biuret reagent. So you might be asked, which of the following will test positive to Biuret reagent? Look for anyone that is a protein. If you don't see any protein molecule there, any protein option. If you see any protein option, that's the answer. If there is no protein option, see none of the above. Ninhydrin test. This test, let me increase the, Ninhydrin test detects free amino groups in amino acid and proteins. Free amino groups in amino acids and what? Proteins. So capture that. Free amino groups in amino acids and proteins. Now, most proteins have a free amino end because every protein must have an amino end and a carboxylic end. So that free amino end will react with the Ninhydrin reagents. Now, the reduced form Ninhydrin test, which is the triketohydrin hydrates, oxidatively deaminates the amino acid, releasing the NH3 or an aldehyde. Now, the reduced form of Ninhydrin then reacts with the released ammonia and another Ninhydrin molecule to form a blue-purple colored complex known as what? Rohmann's purple. You can see that the test tubes, we have phenol, we have thyroxine, we have egg albumin, starch, and distilled water. Now, the starch will not turn purple. The distilled water will not turn purple. Thyroxine will turn purple. But remember, thyroxine will not turn purple in Biuret test, but it will turn purple in Ninhydrin test because it has an amino end, free amino end. Egg albumin is a protein. It has free amino end. And even though it also have some lysine, lysine has a free amino group, which also reacts with the Ninhydrin reagent. But phenol will not react because phenol is just a benzene. Phenol is just a benzene that has hydroxy group. Phenol is just a benzene that has a hydroxy group. So phenol will not react because there is no amino group in phenol. So it might be asked, which of the following will react positively to Ninhydrin test? If you have any option that is just an amino acid, it is correct. They might give you 2 amino acids, give you thyroxine, give you phenylalanine. They are both correct. So you can choose, because there are some questions that will guarantee you that you must pick multiple choice. It's multiple choice. So it might, there's an objective question that have more than one choice answer. So you have to pick the 2 of them. If you pick one, you failed it. You have to pick the 2. So that is that. We have the Xanthoproteic test. For Xanthoproteic test, this detects aromatic amino acid, thyroxine, tryptophan, and phenylalanine. So this test is used to test if there is presence of a benzene ring in the amino acid or in the protein. Now, concentrated nitric acid reacts or nitrates the aromatic ring by electrophilic substitution. This forms a nitro derivative of the aromatic amino acid, which are yellow in color, or which is yellow in color. On adding an alkali, which is sodium hydroxide, the nitro compound forms salt that gives a deeper orange color. So you can see here, the starch will not react. The distilled water will not react. But the egg-albumin, because it's actually a protein that has this aromatic amino acid, so it will react. Thyroxine will react because thyroxine contains a phenyl group, an aromatic group. Phenol will also react. So they might also tell you which of this will react positive or detect or will give a positive indication of Xanthoproteic test. If there's phenol there, phenol will also react to. So you can choose phenol. Thyroxine will also react. Egg-albumin will also react. If they give you phenol, thyroxine, albumin, and option D, you now say all of the above. The answer is all of the above. The answer is all of the above. So we have the Millon's test. The Millon's test detects phenolic group too, like Xanthoproteic test. So it detects phenolic groups of thyroxine. The Millon reagent, which is mercury in nitric acid, reacts with a phenolic hydroxyl group of thyroxine. This forms a red-colored mercury complex upon heating. So you can see that thyroxine has a hydroxyl group. This is thyroxine. Thyroxine has a hydroxyl group. Then this other carbon CHCOO minus NH3 and hydrogen here and hydrogen there. So this is thyroxine. Now, if you remove everything up here, we have phenol. So since the Millon's reagent react with a phenolic group of thyroxine, which is this, that means if you put it in phenol, it will also have a positive indication. So it will test positive to phenol, to thyroxine, egg-albumin, but it will test negative to starch and distilled water. So take down notes, don't miss it. So this is actually one of the take-home questions. Now, a protein is purified by column chromatography and shows a single band on SDS page. SDS is sodium dodecyl sulfate. Page means polyacrylamide gel electrophoresis. So a protein purified by column chromatography. We're going to working with proteins, how we can actually isolate a protein from a milieu of proteins. So a protein is purified by column chromatography and shows a single band on SDS page under non-reducing condition. Under non-reducing condition. This is where I will explain what I was talking about when I talked about insulin. However, when the same protein is treated with the reducing agent, such as beta-mercaptoethanol or DTT, prior to electrophoresis, it yields two distinct bands with molecular weight of 30 kilodalton and 20 kilodalton. Question A, what does this result suggest about the quaternary structure of the protein? One, the protein is not the quaternary protein. Protein is a tertiary protein because the reducing agent is actually employed to break the two yes, let me new slide. Okay, so next slide. So yeah, so now what does it suggest about the quaternary structure of the protein? The protein is not a quaternary protein, it's a tertiary protein. Because here it's telling us that the linkage is a covalent linkage. But if we want to bring in a reducing agent, that means we want to break the covalent bond in the protein. So it's actually is a two-chain protein. It's a protein that has two chains, chain A and chain B, linked together by a sulfur or a disulfide bridge. Linked together by a disulfide bridge. I will explain later. Now, explain the role of the reducing agent in this experiment, which is to reduce the disulfide bridge. What is the most likely native molecular weight of the intact protein? Now let's do some calculation. Molecular weight of the intact protein. The first question is, now listen, if we do gel electrophoresis, we have the gel like this. Now after, okay, let me do this quickly. In column chromatography, we have a column like this. We have a column like this. So here, it contains a sample. If we pour the sample into this place, I will have a beaker here. The sample, I will have beads. We have beads here that will create a path. As the protein migrates, remember, it's a cocktail of proteins. As the protein migrates, they will migrate based on their molecular weight, depending on the type of chromatography. Depending on if it's a size exclusion chromatography, the protein will migrate and the purified protein will be found here. Now, if you want to know if your protein is purified, if your protein is a single protein or is a purified protein with the same molecular weight, you have to conduct the SDS page electrophoresis, which you have a gel. Now, this gel will have wells. It has wells. The wells are the chamber where you inject your sample. If you inject your sample here and you place the gel into an electrophoretic machine, an electrophoretic machine has two sides, positively charged and negatively charged parts. Now, but before you place it on the gel, you must also know the overall net charge of the protein. If the overall net charge of the protein is negatively charged, you place it at the negative terminal. Then when you on the electrophoresis, when you on the machine, the protein will migrate. Let's assume this is where you have your protein A. The protein will migrate and occupy a band, a portion, we call it the band, and occupy a band. That band there tells you that it's a single protein. But if the protein migrates, okay, now this is the question. On electrophoresis, remember the question says a protein is purified by column chromatography and shows a single band on XDS page under non-reducing condition. However, when the same protein is treated with the reducing agent, such as beta-mercaptoethanol, prior to electrophoresis, that means they treated the protein before subjecting it to electrophoresis, it yields two distinct bands. That's what we're seeing here. So this is the same protein A under which have been treated with beta-mercaptoethanol. So they now found out that it has one band occupies here, another band occupies here. So now found out that this protein that we see as a single band is now, after treated with this beta-mercaptoethanol, which is a reducing agent, is now having two bands. One with a molecular weight of 30 kilodalton, and the other with a 20 molecular weight of 20 kilodalton. Now, at the end of the day, we now have two bands. One is 30 and one is 20. One is 30 and one is 20. So that tells us that this protein looks like this. Protein A and smaller molecular protein, protein B. Now the small molecular protein is 20 kilodalton. While the large molecular weight protein is what? 30 kilodalton. 30 kilodalton. Now, before treatment with beta-mercaptoethanol, this is how they look like. So this is how they look like. So it is one protein. So the way it is now, it is one protein with 50 kilodalton. There's one protein with 50 kilodalton. But after treatment, a reducing agent will reduce this. The protein will now look like this. So it is now separate protein. So the protein will now look like, let me delete this. So the protein will now look like this. SH. 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 So the protein will now have two separate proteins, one with 20 kilodalton, one with 30 kilodalton. So this is what the reducing agent did, is separate the two chains. So we have to have separate chain A and chain B. So this is our insulin. So if we are treated insulin in like manner, insulin would have behaved like this. So whenever you see SH, it means that the amino acid at that portion is cysteine. Cysteine. Amino acid at that portion is what? Cysteine. So that is that for today. we'll look at, in our next lecture, we'll look at working with proteins, how we can isolate proteins, but for students of photo, we need to have one more lecture on nucleic acid, nucleoproteins, and juggle some questions under that under that topic. So, and the lecture will be coming maybe 2 weeks from now. 2 weeks from now, we'll have another lecture on nucleic acid so that it will prepare you for your BCH 202 exams. For BCH 202 exams. So under this thing, we'll have on our chat WhatsApp platform, I'll send a question bank so I can actually attempt and assess ourselves. The answers will be released to you before the next class. So we have a question. This is an opportunity for us to bring in our questions. Thank you very much. This is for question time. It's question time. It's question time. Ask your questions now. You can ask your questions now. For those of you that have not liked and subscribed to this channel, please do so. Do you have any questions? What is that? When are we having this 504 lecture? uh, Probably next week. Probably next week. I'll communicate a date to the course rep. So let's ask our questions. At this point, I'm waiting for your questions. If there are concepts that you do not understand, this is an opportunity for you to ask the question. Okay, so we can continue with that question on our WhatsApp platform.